Good afternoon. My name is Nils Stein. I'm working at IPK Gardesleben, and it's my pleasure to be here with you, and thanks to the organizers from the Global Plant Council for inviting me to this panel discussion. I'm talking in the next few minutes about unlocking barley diversity for research and breeding in the context of access and benefit sharing and digital sequence information. If you go to the Genesis website or the Genesis database website, this is a single hub to explore the world's crop diversity conserved in Genebanks, and you query for the term Hordeum vulgare, you'll find 250,000 accessions are present in the Genebanks that have provided information for the Genesis system. The German Genebank here with the code identifier DU146 is represented with 22,909 accessions. So these databases give you access to the genetic resources, but they do not provide you necessarily with enough information about the properties of material. But the diversity that is represented in such collections can be very high. You see here in this picture a representation of morphological diversity in barley spikes close to maturity. And you see that the morphological diversity is pretty different from what you're used to see in barley fields here in Central Europe. The FAO in their second report on plant genetic resources for food and agriculture in 2010 has expressed concerns about the potential impact of climate change, or these concerns are constantly growing, and that plant genetic resources for food and agriculture are recognized as being critically important for breeding or current crops to adapt them to the anticipated future environmental conditions. So we need to be able to have a good educated way to select material for the breeding purpose. But if you look here at this view in this slide, this is typically how collections like at IPK look like. Seed material is stored in cold storage in these jars, and you see, of course, the different numbers. You may even see morphological differences, but how can you really tell which of these 20,000 barley accessions that are in our cold storage are of relevance to your research or to your purpose? Even more, you can grow the material, of course, in the field, and you can evaluate them for different traits. For instance, you have evaluated this material for disease resistance, and you found accessions that are resistant, but can you tell from the morphological difference here how related the material is? So if you select material that is resistant, would you always select material that is almost identical genetically, or it's really divergent and diverse? So we started in 2015 a project we called Barley Bank Genomics, where we really aimed to provide the barley accessions with more information, especially detailed genotype information. So we went to the cold storage, took seeds from every accession, grew a single plant and extracted DNA, and genotyped this DNA with next generation sequencing to generate a high density genotype. This information then was used to analyze the population structure and link the genetic diversity information to morphological trait information that was stored in our records. But I want to give you here only one example. The study was published in 2019, so you can also find all this information in the paper. But if we visualize the diversity information here in this principal component analysis, and we color each individual accession by the geographic origin information where the material was collected or coming from, you see that this diversity space is structured by the geographical origin, with East Asia, Central Asia, Africa, and Western Europe here. So if you have now material that is unknown to you and you generate a similar genotype, you can put it into context, and based on the genotype you can make a proposition where the material originally originated from. So a genotype provides you with very useful information. All the data was made available publicly and is accessible from everywhere in the world where you have access to the World Wide Web. We have submitted the raw data to the Nucleotide Archive at EMBL-ENA, and we have also published digital object identifiers for the different data sets, and we have also provided access to the information through a dedicated web portal, which we call the Bridge portal. Bridge was the name of the project. So this is here the portal that gives you access to all the project information. It's called Bridge IPK-Gardasleben.de, and you can download from here the variation file, and you can also use different visualization tools to get an understanding of the diversity in the population. You can use a lasso tool to mark material or accessions here in this PCA and click on it, and you can then be directly led to the ordering portal of the Genebank information system here at IPK. You will find more information or tools that allow you to generate more information on a material. The genotype information is also very useful to make comparisons between collections and ask the question, is IPK already a comprehensive global collection, or are there gaps in our diversity space? And here we did collaborate with 5 other Genebanks, ICARA, Pavlov Institute, a European project called WheelBee, and the Chinese Academy of Science, Agricultural Sciences in China, and the Swiss Genebank. And the red dots indicate accessions coming from these collections, and they overlap pretty much with the diversity space we have also captured in our Genebank for the respective geographic origin material. So this genotype information is very instrumental to estimate the overlap and the uniqueness of Genebank. We have taken this further now to the European level. We coordinate a H2020 project called Agent, where we collaborate with a number of different European Genebanks to build an actively collaborating Genebank network and ask the question, how related, how unique, how redundant are the different collections of wheat and barley? We use this information also to really characterize and capture the barley species diversity by running a Barley Pan Genome project. We have downsampled the global diversity into core collections, which were resequenced to different sequence level, and we have identified 20 accessions which were sequenced to full genome assembly, and this data was published and it was made public last fall, giving the first insight into the Barley Pan Genome. The material we were working with in this study is barley. Barley is an Annex 1 species of the International Treaty of Plant Genetic Resources and Food and Agriculture, so it's part of the multilateral system for access and benefit sharing. And IPK, the studies we were doing here, all relied on material that we are allowed to provide on the basis of the SMTA. But to really connect our information to the global barley diversity information, we think it's really needed to continue unlocking the barley species genome diversity for research and crop improvement, and the digital sequence information has to be and remain fair, open access, and a public common good. Thank you for your attention.